A new method appears to improve memory in rats, according to a study published January 26 in the science journal Nature. Scientists from the Mount Sinai School of Medicine showed for the first time that a naturally occurring molecule found in the brain during memory formation is helping rats enhance the strength and duration of some types of memories. When the substance, known as IGF-2, was blocked from the brain, the rats didn't remember what they had learned.
And then we went on and asked the question of what does IGF-2, if added IGF-2 in the brain, what does it do to memory formation? And we found that when we deliver IGF-2 in the hippocampus after learning, we see a significant enhancement of memory retention. And we also see a significant increase in the maintenance, in the persistence of the memory. So we basically slow down forgetting.
The findings showed improvement in an important area of memory known as declarative memory, the ability to remember places, facts, and things. Declarative memory is affected in Alzheimer's disease and other forms of dementia, and researchers have long sought ways to improve or preserve this ability.
One interesting advantage of this result is that IGF-2 passes the blood-brain barrier. And the blood-brain barrier is this protective structure that protects the brain from the circulation. So, for example, hormones released in circulation, or toxic substances, or a number of exogenous outside things that get into the circulation are not going to get into the brain because the barrier blocks them. But what the barrier does also blocks most of the proteins, most of the peptides, and most of the drugs. So it's very difficult for drugs to target brain function because of the barrier. But IGF-2 passes very easily the blood-brain barrier, so that's a great advantage.
In the study, the rats were placed in a 2-compartment cage where they would receive a mild foot shock when they stood at one end. They quickly learned to avoid that area.
The measure of our memory is actually a latency, an avoidance to this place in which they experienced something unpleasant before. And so the strength of the memory is measured by measuring the time that they avoid this place. And for example, the animals that have been treated, the control animals, have in our conditions have a latency of about average 300 seconds. The animals that were treated with IGF-2 had a latency that was around 700 to 800 seconds. So a much stronger avoidance of the place.
The effect was specific for certain parts of the brain. IGF-2 also appeared to boost memories only during a specific window of time, when they were being formed initially or when they were being recalled. The next step is to administer IGF-2 to the entire body to see if it can produce the same memory-boosting effects as direct injection into brain regions. It is too early to say whether IGF-2 will be useful in humans, but the substance may hold more promise than molecules that have been studied up to now.
We obviously need to do the experiments. However, we hope that this is very applicable to humans. And two reasons make me say that. One is that the structure of this protein is very similar in different species, animal species, and from the more simple to the more complex. And the regulation of this protein in the hippocampus during memory formation occurs within a regulation of proteins that is highly conserved throughout different species, from very simple ones like Drosophila, which is the fruit fly, or sea snail, Apresia, up to humans.
For the Wall Street Journal, this is Shirley Wong.